hear me okay? Okay, great. Thank you all for being here on this beautifully sunny afternoon. It was probably a tough choice, do something outside or come in here. So I'm glad you're here with me today. And I'm really humbled to be selected for this lecture. I just was so happy to get the invitation. That spelled out that Rose Litman was a rigorous scientist. She also sounded like someone who lived a really fun life. And she did a lot of mentoring of others. And this is something that's very near and dear to my heart as well. So I'm going to save maybe 5 minutes or so at the end of my talk to talk about mentoring. But first, I'm going to talk about some of the science. So a lot of times people ask me, why should we send humans to space? This is a big part of my research. How does being in space change humans? And so I get a lot of these kinds of questions, like why don't we just send robots or rovers? Sending humans to space is very expensive and it's dangerous. And I always think about this quote from astronaut Bill Anders. This is his iconic Earthrise photo that he took. And he said, we came all this way to explore the moon, and the most important thing is that we discovered the Earth. And I really feel that we have learned so much about our history, our biology, geology, et cetera, by sending humans into space. You're probably in agreement with me if you're here, but just that's a question that I often get. And I was really fortunate as a graduate student to have a NASA graduate student fellowship. This allowed me to go back and forth to Johnson Space Center to work with folks there in the neuroscience laboratories, some of whom are still my collaborators today. We won't say how many years ago I was a graduate student, but I can tell you 20-some years ago now, I was there for a summer faculty fellowship. And that's when I first started talking with my colleagues there, and we said, I wonder how going to space changes the human brain. You know, around this time, we were learning a lot about neuroplasticity, how the brain changes when you learn a new skill, or you learn a new language, or to play a musical instrument. And we thought, wow, what an awesome opportunity it would be to look at how the brain changes when people go into space. We're talking about putting people in an environment that is outside of our evolutionary history, right? So how would we respond? How would the nervous system adapt to that? And so during that summer, with my colleagues, I wrote a grant proposal to study this, and a call for proposals came not too long after this that we thought was appropriate. We submitted our proposal, and it didn't get selected. And you know, I try to remind myself of that experience sometimes and say, oh, we were just a little bit ahead of our time. But we got some weird review comments. Reviewers asked things like, why would you study how the brain changes in space when there's no evidence that the brain changes in space? And we thought, no, that's what we want to look at. But I have learned over the years that when you get a paper rejected or you get a proposal rejected, it doesn't mean no. It just means not now, okay? So this is maybe a lesson in persistence. A couple years after our proposal was not selected, out came another call for proposals that basically said, we want people who will study how the brain changes in space. And so, you know, we took this old proposal and dusted it off, and we changed almost nothing. We changed, you know, updated a few references, put it in, and got selected. And, you know, I feel very fortunate that we've been able to do this work. It's been very exciting. And, you know, again, these are ideas that we had starting maybe 20-some years ago. Some of our ideas then seem a little maybe outdated to me, but we really thought that we would see evidence for neuroplasticity and sensory motor adaptation. And I'll show you some data in support of that we did. But we also saw some really unexpected things. And in hindsight, I don't know why we didn't expect to see some of these things, but I'll also share some of those findings with you. So when we started, we, of course, already knew there had been a huge, rich history of behavioral research with humans and non-human animals coming back from space. And we already knew that people, whoops, what did I do? There we go, wrong button. We already knew that people would come back with declines in mobility and balance, and various animal models had also been studied in space. So we knew there was some evidence for changes in central nervous system structures that were involved in sensory processing and motor control, but this hadn't yet been looked at in humans. So we were very excited to kind of leverage spaceflight as a sensory motor adaptation and neuroplasticity laboratory, okay? And again, if you think about our evolutionary history and our anatomy and physiology, a lot of our systems have evolved to either work against gravity or to actually leverage gravity, make use of gravity in signaling. So just one example, if you think about the heart is pumping blood upwards against gravity, right? And we have lots of specialized valves in our circulatory systems that if we didn't have them, all of our body fluids would be in a puddle in our feet, and we would look very weird, okay? But these systems have evolved to work against gravity. There are also systems that have evolved to leverage gravity. Before I tell you about one of those, I'm going to give you a pop quiz, okay? Audience participation time. Who in this picture is right side up and who is upside down? Nobody. That's the right answer. Nobody. Okay. And raise your hand if you've heard of the vestibular system. Almost everyone. Okay, good. I think most people know about the cochlea, this snail-like structure in the inner ear that's involved in hearing, but we also have right next to it our vestibular system, right? So these semicircular canals that detect angular accelerations of the head, and then this kind of blob structure called the otolith, which is involved in detecting linear accelerations and angle of tilt of the head. And I'll show you on the next slide that it does that by leveraging gravity. So if we put people up in space in microgravity, there's not a signaling mechanism for them to detect how their head or their body is tilted relative to gravity, a straight up and down gravitational vector. So these people may not have any sense of which way is up or down. Sometimes astronauts have illusions that they're right side up, and then suddenly they feel like they're upside down. So this system is really not working in the absence of gravity. So this is how it works. We have these calcium carbonate crystals that are sitting in this, on top of this kind of gelatinous goo in the otolith. And when you tilt your head, the force of gravity pulls those stones across these hair cell receptors, which mechanically causes them to open ion channels. They depolarize, send action potentials to the brain, telling you you've tilted your head. So here's a little more audience participation. If you all would please humor me and take your right ear and tilt it towards your right shoulder. Okay, so obviously your visual cues tell you you've tilted your head. You also have proprioceptors in your neck muscles and tactile sensors in your skin, but also your vestibular system is saying, hey, your head is tilted. Okay, you can stop doing that. But now imagine you go to the International Space Station and you're in microgravity and you tilt your head. You still have vision saying, yes, your head is tilted. Your neck muscle and tactile sensors are saying, yes, your head is tilted. And this system is saying, no, guys, nothing happened. And so the brain is getting very conflicting sensory inputs. This is what can lead to space motion sickness, right? And so we were really interested in studying how the brain processes these vestibular inputs and how it changes from pre to post flight. There's behavioral evidence that the brain sort of stops processing these signals in flight. They're unreliable. And so the brain starts ignoring them and focusing more on vision and proprioception. So our proprioceptive systems are also altered in microgravity, right? So look at how these guys are moving around in their environment. That's not how you got here today, right? As I'm standing up here, I don't even think about it, but I have these large anti-gravity muscles in my legs that are contracting so that I don't just crumple on the floor. They're supporting me against the effects of gravity. These guys are not engaging those muscles. In fact, if they want to travel from one end of the room to the other, all they would have to do is just tap a finger against the wall and they could float to the other side. So when you are in microgravity, you also have reduced inputs in terms of these skin receptors. You're not walking around on your feet. You're not standing on your feet. In fact, your clothing is not even touching your body. It's floating over your body. Some astronauts come back from space and they complain that their skin hurts for a while until they get used to activation of these touch receptors again. And I've been talking about these proprioceptors in your muscles as well. You have them in muscles and tendons that tell you where your body is in space. And again, if you're not loading your muscles in the same way, you're also not getting the same inputs from that system. So again, there was already some behavioral evidence that was accumulating more and more while we were collecting our data. People adapt to this in flight, and you can see it evidence as after effects when they come back. So these are data from my collaborator, Scott Wood, at Johnson Space Center. He tests a lot of crew members pre and post flight using this crazy balance system, okay? This is actually a really hard balance test. If you see, this woman is standing on a plate, and you can change the system so that as she's leaning forward, that plate will also tilt forward, meaning that as you're leaning, you're not getting proprioceptive cues from changes in your ankle joint angle, okay? So if you do this with your eyes closed and while moving your head, it is really hard, okay? It's challenging. So these data, the blue and the green are from shorter versus longer duration astronauts. But these are the pre-flight measures where a perfect score is 100. And you can see that even pre-flight, people are not very good at this test. It's hard. But if you look at where they are in the initial days when they first come back, they are even worse. There's a lot of data points that aren't shown here where people just cannot maintain their balance on this plate under these conditions at all. It takes them time to readapt. It can take up to 10 days or so to get back to where they were pre-flight. And we think this is because, again, when you're in microgravity, your brain is suppressing these unreliable vestibular inputs. So when you come back to Earth, you're getting all these vestibular signals again of head tilt, and you don't know how to do the sensory integration to maintain your balance. So our idea was, pre and post flight, we would get lots of measures of balance and mobility and cognition. This is actually astronaut Kate Rubins on the International Space Station doing one of our cognitive tests while she's up there. And then we took lots of different brain measures pre and post flight as well. And our idea was, can we map these behavioral changes to any brain changes that we see in a way that might suggest that there's neuroplasticity or some kind of sensory reweighting, suppression of vestibular cues that is taking place. This was our data collection timeline. So we measured people 6 months and 2 months before they went to the International Space Station. We had them do a few of our tests while they were up there. There's no MRI scanner up there, obviously, so we couldn't do the brain scans while they're there. But we measured them 3 times, kind of early, middle, and late in the mission on various cognitive and motor tasks. And then we catch them within their first few days of return, and we follow up out to 6 months post-flight. So this is, for a single participant, it's 1.5 years of data collection. under the best of circumstances. So how long do you guys think it would have taken us to collect our data set with 15 astronauts? How long do you Any ideas? I hear some very quiet rumblings. 2 years. 2 years? Almost a decade. Almost a decade, right? Because we're not sending people to space, to the International Space Station like every month, right? It's a few people a year. They're not all in the same experiments. So this took us a long time. And by the way, part of that decade was during the pandemic. And we had some really wonderful collaborators and staff at Johnson Space Center who put themselves in quarantine so that they could test our crew members when they came back, having never been exposed to COVID before because there was outbreak while they were on the space station. So we did our best to keep our quarantined investigators happy. We sent them cookies of a moon landing or Blue Moon cookies or M&M rocket cookies. And in the end, we made it through, but it took us a really long time. Okay, so again, we hypothesized that we would see some evidence for neuroplasticity, especially in this part of the brain that processes vestibular inputs. This is what we call the vestibular cortex, or in anatomical terms, it's parietal operculum 2, but I would just call it vestibular cortex. And we did see some evidence for this, but we also saw some evidence that it took us a while to figure out what it was, and it was actually evidence for direct mechanical effects on the brain. And I'll explain what I mean by that in a few minutes. Let's first look at the neuroplasticity changes. So one of the things we looked at was we used a particular MRI sequence that allows you to look at white matter tracts in the brain. These are large bundles of axons. You can think about them as like brain network highways. They're big connections between different nodes in a network. And we're able to quantify the integrity of those white matter pathways. And what we found was white matter changes in the cerebellum, which is a structure that processes sensory inputs and controls movement. We saw white matter changes in this region underlying the vestibular cortex, and changes in the corticospinal tract, which is this pathway connecting the motor areas in the brain with the motor neurons in the spinal cord, telling the muscles to contract. And in all of these cases, these white matter changes were similar to what you would see with aging, but accelerated by 3 to 4 times. So in other words, this is like white matter atrophy in pathways that are involved with contracting muscles, which I told you doesn't happen as much in the absence of gravity, right? Pathways that are involved in processing of vestibular information. But if you're getting unreliable or no inputs from part of that system, you're probably going to get atrophy from disuse. And same thing in the cerebellum, reduced white matter integrity. We found as well that this is not the tightest correlation, but it was statistically significant where people who showed the most, crew members that showed the most declines in white matter integrity from pre to post flight in this vestibular cortical region had the worst balance when they came back, okay? It's almost like maybe they don't know how to re-engage this system initially, or they might need longer to readapt. We also wanted to measure, and sorry, I forgot to say, this work was done by former postdoctoral fellow Jessica Lee. And so we also wanted to study brain functional responses to vestibular stimulation. And I've told you that the vestibular system detects accelerations of the head, right? But you can't accelerate the head in the MRI scanner. People have to lie extremely still. Even heartbeat and breathing move the brain around on the scale at which we're measuring. So we couldn't do head accelerations in the MRI scanner. Instead, one of my former PhD students, Fatima Nui, did a lot of work to help us kind of engineer and validate this system, which relies on compressed air to control a little motor that we use to stimulate the vestibular system, and we measure responses in the eye muscles. And she was able to validate this with some studies on aging and comparing to other ways of imaging the vestibular system. So then, another former PhD student, Kathleen Hupfeld, went on to look at vestibular responses pre- and post-flight in astronauts. And this is what brain activity looks like pre-flight if you apply this vestibular stimulation. So this vestibular cortex that I mentioned here is activated when you stimulate the vestibular system. So is part of the cerebellum. And sorry, for people that aren't used to looking at these, this is a side view of the brain. The eyes would be over here. These two are if you were looking at the brain through the nose. And this one is just an extraction of the cerebellum. Now, look at all these other areas, though, that are in blue. Like, what the heck is happening there? These are areas in the back of the brain that process visual inputs, or here, that process these proprioceptive and tactile inputs. And this is very typical if you stimulate one sensory system. So if you were in the scanner listening to auditory sounds, you would activate your auditory cortex. Other sensory systems would be suppressed. It's kind of like the brain is saying, hey, something's coming in here. I got to pay attention. I'm going to activate the regions that are important for processing this signal, but I'm going to tell some of these other ones to quiet down, okay? So that's what we see pre-flight. And if you keep your eye on these blue areas, let's look at the areas that change after crew members come back from the International Space Station. It's the same areas. So these visual and somatosensory proprioceptive processing regions, which pre-flight were suppressed, now post-flight are actually responding to vestibular stimulation. So these are our group data points. These are the first two pre-flight time points. Again, these values are negative. They're less than 0. So pre-flight, these are suppressed. First day that we measure them, this is about 4 days after they've been back, the values are now positive. So these brain areas are responding to vestibular inputs. And it takes at least a month, maybe about 3 months, to get back to where they were pre-flight. To try and understand this better, we looked at individual differences in this change and whether it related to individual differences in balance changes. And the answer is yes, it does. So this is an area in the somatosensory cortex, which processes proprioceptive cues. And these are single astronauts showing their sort of change score from pre to post-flight. We've got increased activation going up on the y-axis of this area and better balance going to the right on the x-axis. So people who activate proprioceptive parts of the brain in response to vestibular stimulation more have better balance post-flight. So suggesting that their brain has kind of realized, vestibular cues, no good. I got to start paying attention to other sensory modalities. Same thing happens with some regions in the visual cortex. People who activate these areas more after you've been to space have less balance declines when they come back, okay? So we think this is support for sort of neuroplasticity of sensory processing and neural evidence for sensory reweighting from pre to post-flight. So again, if you're in microgravity, you have these altered vestibular signals coming into the brain. And the brain seems to respond by saying, you're not reliable, I'm going to ignore you. And I'm going to pay attention to these other sensory cues and sensory systems. And then we think post-flight, there's some lingering of this for, we don't know exactly how long, maybe a few weeks to a month. And that seems to relate to how you recover your balance when you come back, okay? So for us, this is kind of interesting scientifically, But it's also interesting in terms of applications. If you think about where we're going next with humans in space, we are sending people back to the moon. We are maybe taking people to Mars. And I showed you the picture of what it looks like when people come back from space to Earth. They've got a whole team of people there. They put the crew member on a litter. They're carrying them around. They connect very quickly with their team of physical therapists and athletic trainers. That's not going to happen when people show up on Mars. Those people are not going to be there. So if there are things that we can identify where people have better balance kind of right out of the gate, we can think about ways to leverage that so that when we put people on Mars, we know that someone can go right out and drive the rover, or if there's an emergency, they can open the hatch and get everybody out. And so we think these are also interesting in terms of thinking about different applications. So what I've shown you so far is some evidence that we found for neuroplasticity, right? Some of this disuse of systems results in some atrophy. Shifting reliance on sensory systems also changes how we process sensory information when we get back from space. But I also said something about physical changes that we were surprised by. And again, in hindsight, I don't know why we were really surprised, but we were. We initially didn't even know how to interpret these findings. So I talked about all these fluids being pumped up in the body against the effects of gravity. So it's been known for quite some time that those systems are still working, right, when you go to microgravity, but they're not working against gravity. There's no more gravity pulling stuff down. So when we first go into space, this head is much bigger, right? You can even see photos of astronauts when they first arrive at the International Space Station. Their faces are puffy. The upper body is kind of puffy. You get this very large fluid shift. The legs look very thin. This has a very technical name. People call it puffy face chicken legs. But this happens, right? And over time, you equilibrate a little bit if you're up there long enough. But, we really should have paid more attention to this big head here and thought, where is all that fluid going, right? And I can tell you where some of it is going. Some of it is going into the cranial compartment and into the cerebrospinal fluid-filled spaces in the brain. So I am going to show this little animation. And obviously, this is where I want you to look. I don't know how you would know that. But this is the top of the brain. And you can see this black right above it. The black is fluid. It's cerebrospinal fluid. And this inner white line here is the skull. This is actually skin and fat. This is the skull. So this image is going to toggle back and forth between pre and post flight. And I want you to keep your eye on this dark space. So look at that. The brain is shifted up. And that fluid is getting squished out of the way. The brain, in some places, is physically touching the skull, okay? It's getting a little bit compressed. And again, if you're not used to looking at these, sorry, this is a side view. Eyes are over here. Okay. We also see a lot more fluid in a structure called the ventricles. This is a system in the middle of the brain that is filled with cerebrospinal fluid. So this is a side view, and this is a view as if we were looking in from the top. So we have lateral ventricles on both sides of the brain. And this red dashed outline was created from an algorithm that we use to segment out the borders of those ventricles. So this is where it is pre-flight. This red dashed line will stay here. And where I want you to look is around the edges. And you will see that this black expands beyond the borders when we get to post-flight. So you can see along the top here. You can see a lot here in the front and along the back. And in some crew members, these changes are fairly small, maybe 5% increase. In others, we see 25 and approaching 30% increase in volume of these structures. And it's not just that you have more fluid here, right? Like, look at this. These are much bigger, and stuff has to go somewhere, right? They're compressing the surrounding brain tissue, okay? And we did manage to get a hold of some retrospective MRI scans from people who flew on shuttles. So on this graph, we basically have change in ventricular volume in milliliters. These are pretty small structures, so even a milliliter of change is fairly big for this system. And the x-axis is current mission days. So if we look at this first data point, these are people who flew on shuttle. So they were up for a matter of days to weeks, and really little change or no change in ventricular volume. Then these next data points are people who spent 6 months on the International Space Station. And then this next one, this last point is only a sample size of 4. These are people who spent about a year in space. So first thing we can see, like if you're a space, you're going on a space tourism flight right now, you're going for hours or days, I would say don't worry about this. If you're going for 6 months or a year, I would say maybe I would be a little worried. But it's also reassuring to see no additional increase from 6 months to 1 year. Again, very small sample size for 1 year. It's only 4 people. But if that line continued to go up, then I would say, what if we send people to Mars and back and it takes a few years, their whole head is just going to be a giant ventricle. There's going to be no room for anything else, right? So kind of some pros and cons here of what we found. We also found that it looks like it takes maybe somewhere between 1 and 3 years for these structures to recover. We don't really know for sure. And it may be that recovery might mean shrinking back down, or it might mean recovery of elasticity of this structure. So it might expand and then not be completely fluid-filled, but just not rebound initially when people come back. So I think there's still a lot of questions here. We also used a particular MRI technique that allows us to look at fluid in the brain. It's typically referred to as free water. It's not just water. There's other like neurotrophic factors and things like that. But this allows us to look at fluid shifts within the brain tissues themselves. And what we see is a lot of blue here at the top of the brain. And that reflects what you just saw on those alternating images, that the brain is sitting higher, the fluid is just getting pushed out. And it has to go somewhere, so it is found more along the base of the brain. We also found that some of these changes vary depending on whether this is your first time in space, your second, your third, or your fourth time. You have sort of even different direction of effects, so we don't really understand what's happening here. One thing I will say, though, about the ventricular volume changes and the fact that they don't seem to happen for short duration flights and they don't recover very quickly. I actually think in some ways that's kind of a good thing. Like, I don't think we want people to come back from space and have all the fluid gush out and their brain immediately thunk back down into place, right? So, I think there's something maybe sort of adaptive about the slow nature and slow recovery of these changes. But again, we don't really understand all the implications yet. So to summarize the research part, again, I'm going to spend a couple minutes at the end talking about some mentoring stuff. But to summarize the research part, what I have shown you is that there are changes in both brain structure and brain function that occur when people go to space. Some of these changes seem dysfunctional. We don't really have a good handle yet on health implications, long-term health implications of some of these fluid shifts. But some of these changes seem very adaptive. If you're getting unreliable sensory signals, why would you pay any attention to them? And so some of this seems very adaptive. And if you're interested in knowing more, we together with some of my trainees and a great collaborator, we published a review paper a few years ago that has almost the same title as this talk, where we really kind of lay out what we think is happening and what different predictors, lifestyle factors, et cetera, might be that interact with these. And then last year as well, I published an article in The Conversation, which is like kind of like a popular press type outlet about risks of human spaceflight. And also last year, I gave a talk about more of my research, a webinar on The Scientist, which is also really aimed at broad audiences. So if you guys want to learn more, you can go ahead and take a picture of those. Maybe I've talked long enough for everybody to get a picture. No, a couple more phones still up. OK, all right. A couple more phones. All right. Everybody good? OK. So what's next in this area for me? Well, I think, as someone who's really interested in how people learn, how the brain changes when we learn, I'm really kind of fascinated by this interplay and co-occurring dysfunctional and adaptive changes. They probably even feed into each other, right? I mean, we know the brain remodels even as we age to help counteract and compensate for some of the declines that occur with aging. And so as this dysfunctional stuff is happening, I'm sure the brain is trying to adapt and figure out new ways to do things as well. So this is something I'm really interested in continuing to study. I'm also part of a new study, the long-term health study. This is a study that has lots of investigators on it, and we are studying brain and ocular changes with human spaceflight, and we are following people out to 5 years post-flight. And NASA told us they think it will take us until 2033 to finish collecting data. So if you want me to come back then, I'm happy to. We also do some of these analog environment experiments. NASA loves head-down tilt bed rest as a model. It does mimic a lot of the body unloading and the fluid shifts. And so I think in some ways it's actually really helpful for better understanding the changes that we're seeing. And we're wrapping up a kind of a slew of papers from a bed rest study where people also got artificial gravity. So they were in bed rest for 60 days, and different groups got different doses of artificial gravity by being spun on a human centrifuge for half an hour a day or not. And there were some interesting effects there as well. And again, before I talk about the mentoring stuff, I want to just close with a huge shout out to all of the hands that contribute to this kind of work. These studies are really difficult to do. They take a ton of time, a lot of effort. And I've had the pleasure and privilege of working with some amazing collaborators and some really wonderful trainees. Vincent Koppelmans, Jessica Lee, Heather McGregor were all postdocs in my lab at one point. And Fatemeh Nui and Kathleen Hupfeld were PhD students. And they now have moved on and are changing the world in amazing ways. I'm very proud of them. But since I learned about Rose Litman, that she really had a passion for mentoring, I wanted to talk a little bit about that and just share some of my experiences as both a mentee and a mentor in science, starting with maybe as a mentee. So I'll tell you what made me really passionate about being a mentor. And it was from a couple of uncomfortable experiences that I had. So when I was a postdoc, my postdoc advisor and I, we published a paper in Science. And for me, like, that's the pinnacle, right? This is like the best journal that I think I could ever publish in. And I was a postdoc. I was on the job market. I thought I had it made. I thought I was it. I got invited to speak at a conference in Spain. I was like, yes, I will go. This is amazing. I can't wait to share my science and see what everybody thinks. And I got to the conference, and I looked at the program, and I realized the entire conference, I would be the only woman speaking at the podium. And suddenly, I felt really small. I felt like now, not only was I representing my science, I had to represent my whole gender. Like, the pressure felt ridiculous. And I can tell you this still occasionally continues to happen to me. And I know that these feelings, of feeling like, oh my God, there's nobody up there like me, are even worse for people who have intersectionalities of race, ethnicity, gender, physical abilities or disabilities, sexual orientation, et cetera. And so a few years ago, the Translational Research Institute for Space Health did a self-study. This is a funding agency for people who work in space health. They did a self-study of who they were funding. And they realized, just like the NIH, the NSF, other organizations, they were not funding women and minorities to the extent that they wanted to be. And in their self-study, they also realized that in part this was a pipeline issue. People were not applying to the same that, if you looked at representation in different jobs and different careers, just people felt some barrier to application. And so they came out with a call for proposals for diversity partnerships. And I said, great, this is my chance to hopefully make it better for some other people. And so I was fortunate that my colleagues at UF, Josephine Allen and Christine Wegner, and my colleague at Texas A&M, Ana Diaz-Artiles, all said yes and put in a proposal with me. We had amazing support staff, Alexis Ganshim and Christina Hall. And we spent a couple of years with this support. We offered grant writing workshops, networking events, mentor-mentee connections. And I think this ended up being pretty successful. We had mentees that submitted 57 proposals just to Trish or NASA. If you count all the NIH, DOD, et cetera, people applied for almost $90 million worth of funding. And to be honest, I don't think we did very much for them at all. They were already amazing. They had fantastic ideas. They just needed someone to just turn them slightly and say, here's where you need to go. Here's the website. Here's what that means. Here's how you navigate this stuff that's impossible to understand if you don't have someone telling you how to do it. So the mentees that participated were all amazing. And a last plug here for mentors. So there were people we asked to be mentors or subject matter experts who said, I'm so busy, I just can't. And of course, that's fine. But we also had a lot of people that we asked who said yes. And we surveyed them after their participation. And they said the program was amazing for them, too, because they found these undiscovered yet hotshot amazing new collaborators. And they were suddenly submitting proposals with the mentees and writing papers with them. And they felt enriched, and they felt like their science got better, too. So I would just encourage all of you to think about mentorship. And if someone asks you, try not to say you don't have time, unless you really don't. Some people don't, right? Some people are busy changing the world in other ways, and that's great, too. But just a little shout out to mentorship and the value that it can bring to our fields of study. So thank you.